All right. Good morning, everybody. Today, I'd like to talk about how myosin is regulated, how cells generate retrograde actin flow at their leading edge, and to explain the clutch hypothesis, which is one of the central sort of models for understanding how cells crawl. To start off and talk about the regulation of myosin, I think that a really good place to start is with skeletal myosin. So a really good place to start to understand the regulation of myosin is with skeletal muscles, and this will help contrast how myosin is being regulated in all other cells in our body. So to begin with, so a muscle cell can be surprisingly long. So it can be somewhere in the range of 1 to 40 millimeters in length. So that would be like 4 centimeters. And it's about 10 to 50 microns in width, which is kind of the average diameter of a cell. One thing that allows muscle cells to be so huge is they're multinucleated. That is to say that a single cell contains multiple nuclei in it, and having multiple nuclei allows a lot of protein synthesis because a cell can generate many RNA transcripts in order to support the creation of new proteins. If we zoom in on a myofibril, we see that the fundamental unit of organization in muscles are the sarcomeres. These, in contrast, are relatively small. So they're about 2 microns long, and they can contract by about 70%. So in our muscles themselves, we've got our muscle cells, and then within those muscle cells are the sarcomeres. That fundamental unit can contract by 70%. So when you think about your muscle contracting, that is the sort of limit of contraction, the contraction that can occur within the sarcomere. And the borders of the sarcomeres are defined by these Z discs, which I'll just talk about in a second. And if you sort of overlay a cartoon of the sarcomere with the electron micrograph, you can start to understand how things are being organized at the molecular level. So we've got our Z disc here on the side, and we have our actin filaments that are attached with their myosins on the Z disc. And then in between are what are called thick filaments of myosin. So these are many myosin molecules that have been bundled together into a long fiber. So when we look at this electron micrograph at the top, these sort of dark bands here, those are the Z disc, and you can see these darker lines in the center. So these are the myosin filaments. So, and generally people call the actin filaments thin filaments, and the myosin filaments thick filaments, and it's just based solely on their size. So, as I mentioned, the thin actin filaments are assembled with their plus ends actually embedded in the densely staining Z disc, so I flipped that around. My apologies there. And then the thick filaments are made of bipolar myosin. And actually, one way to actually think and remember that is that myosin wants to walk towards the plus end of an actin filament. So if we have all of our myosin heads here, and the plus ends of the actin filaments here, these myosin heads are going to be trying to walk to the Z disc. In terms of what regulates the contraction of myosin, calcium is an incredibly important molecule, and of course ATP is needed to provide energy. So if one takes an isolated muscle and adds solutions that have ATP and calcium, it can lead to the activation of myosin and then the contraction of the sarcomere itself. And because the myosins are trying to walk to the plus ends of the actin filaments on both sides, what that does is it brings the Z disc together and it drives contraction. So a couple of things that we want to talk and think about it is, why is it important that myosin 2 is a non-processive motor and has a short duty cycle? And then another question is, what would happen if myosin 2 was replaced by myosin 5 in the muscle? And as just a little bit of a reminder, so in the last lecture, we talked about the biophysics of myosin 2 and myosin 5. And one of the major differences between these motors is that myosin 2 takes really short steps and it can't maintain force. So it takes a short step, it pulls an actin filament, and then it releases it, and then the actin filament can either slide back or do something else. Where myosin 5 is taking these large steps, and it's processive. It's holding on in between steps and moving forward. And just with these two ideas, I'll just kind of set up the question. If we actually had myosin 5 in our muscles, how would that change the process of muscle contraction? Yeah, go ahead.
It would make it more intense.
It would be more intense. So can you be a little more specific though?
Yeah, so because it takes longer steps and it has a greater grip, every time there's movement of the myosin filament, your actin might, I don't know whether it can degrade or it might be too strong for, yes, too strong.
So the too strong is kind of getting at part of the problem. So one thing about muscle contraction is you need contraction and then you need relaxation. So it's important that the myosin can be disengaged from the actin filaments to allow muscles to expand. So because myosin 5 is processive, it would always be gripped to the muscles. Now, if you were in the expanded state, you would have an advantage in one sense in that contraction could be really fast because you'd be able, the myosin's taking large step size, but it would be more of a one-way sort of process where the myosin's stepping to the ends of the actin filaments, but doesn't have the mechanism to let go and let relaxation to occur. So when we start to look at muscles, there's a lot more than just these thick myosin filaments and these thin actin filaments. So muscles are a structure, and as a structure, the assembly of muscles needs to be very carefully regulated. And several of the proteins that we've already talked about are important for this process. So in particular, CAPZ is a protein that binds onto the plus ends of actin filaments and blocks polymerization. It turns out that in muscles, CAPZ is used to attach the plus ends of actin filaments to the Z-disc. And then in addition, there's tropomodulin, which is binding to the minus ends of the actin filaments, and it's preventing their disassembly. So the cells are using different proteins to regulate the length of the actin filaments. And then in addition, there's a couple of other proteins that are just important for the structural integrity of muscles. Titin in particular turns out, well, titin in particular is important as it bridges the different Z-disc here, and it turns out that titin is one of the largest molecules or proteins that people have ever discovered. That's part of the reason that it's called titin. And more or less, it's holding the Z-disc together and preserving the structural integrity of the muscles. And yeah, so we have all of these different sort of proteins holding the actin filaments together, holding the whole structure together. And in terms of sort of putting this into motion, if we sort of zoom in, so here we have a muscle cell, and it has multiple nuclei in it. And then in the muscle cells, we can see that we have our Z-disc here. So these are going to be about 2 microns long. And then in each one of these sarcomeres, you've got the thin and the thick filaments in between them. So in terms of what drives muscle contraction in our bodies is that there's going to be a communication with a nerve cell that's going to release neurotransmitters, and it's going to open up channels that allow calcium to flood the interior of the cells, and then that high calcium activates muscle contraction. And how it does that is it causes the change in conformation of two proteins that are laying along the side of the actin filament. And I guess, like, if you think about it, like, normally you would, like, if you want to drive muscle contraction, what would be the basic mechanism that you think cells would use? Do you think cells would want to directly turn on myosin 2?
What are the options? Either myosin 2 or what?
Well, so skeletal myosin 2 is the motor that drives muscle contraction. And the question is, so when cells need to contract muscles and turn on myosin, what would be the most logical way that you would turn myosin on?
ATP, using ATP.
You would have ATP there, but the thought is that you would somehow directly turn on myosin, probably. But it turns out that muscle contraction isn't regulated by turning on myosin. Instead, what happens is these proteins are preventing myosin from interacting with actin filaments, and then when they move out of the way, muscles can contract. And it's a little bit of a counterintuitive idea here. This is showing sort of a diagram. We've got our actin filament. It's got a helix, so you can sort of see it's rotating. And then we have essentially the motion of the tropomodulin. So as you can see, before the tropomodulin is sort of right here, and then when calcium enters into the cell, it moves over a little bit and it exposes these myosin binding sites. And when the binding sites are present, then myosin can be active. So kind of the default state in muscle cells is that myosin always has the potential to generate contractile forces, and it's being prevented from doing so until calcium comes in and causes these other proteins to move out of the way to allow muscle contraction. And as just a final thing, this is called thin filament regulation because it's regulation that's happening at the level of these actin filaments. And just to kind of put this in a diagram to show how this all works. So calcium's binding troponin, troponin's changing its conformation. These myosin binding sites get exposed, muscles contract. So when calcium decreases, the conformation gets changed, these binding sites are hidden, and the muscles relax. Okay. So it turns out that in non-skeletal cells, there is something called non-muscle myosin 2. And it's the more evolutionarily ancient of these two types of myosin 2. So all of our cells have non-muscle myosin 2 in it. And it turns out that it uses a radically different pathway to modulate myosin activity. So in our smooth muscles as well, calcium causes muscle contraction. And the way that it works is that high calcium causes the activation of myosin light chain. And so on the neck of myosin, there's the essential light chain and the regulatory light chain. And what they're doing is they're stabilizing the neck. And when myosin light chain kinase gets activated, it leads to phosphorylation of myosin light chain. And then that stabilizes the neck of the myosin and it causes its activation. So you can sort of see here, we've got our myosin light chain here. We've got a myosin light chain kinase. We've got our calmodulin. So what's happening here is calcium's coming in. It's causing activation of this kinase. The myosin light chain gets phosphorylated. And then on the other side of this kinase, there's a phosphatase, and the phosphatase is removing these phosphates, and it's causing relaxation. So in terms of the regulation of non-muscle myosin 2, there's going to be a kinase and a phosphatase that are modulating the phosphorylation of this myosin light chain, which causes myosin to be active. One final sort of note, and I'll just throw this out there. Why would you think that there might be two different mechanisms for regulating myosin 2, and what would be the advantages or disadvantages of each? Yeah.
So just in case one pathway fails, another pathway can work to keep the process.
It could be redundancy, so that's a possibility. But there's also sort of different jobs that are needed in these cells. So, for example, in muscle cells, like there needs to be a very rapid response to the signal to contract, and then the cells have to be able to very rapidly stop contracting. And that thin filament regulation allows that, because essentially the myosin's all ready to go, it's activated, but one protein just slides out of the way and it causes its activation. So that's going to be really rapid. In terms of regulating myosin using kinases and phosphatases, one sort of trade-off with that is it's a much slower process. So for myosin to get activated, each individual myosin light chain needs to be phosphorylated. And then for it to be inactivated, each individual light chain needs to be dephosphorylated. So it's a much sort of slower process. So the big advantage with the thin filament regulation is speed. With this, there's finer control at the subcellular level. So cells can control where kinases and phosphatases are.
So these are for two different types of cells, right?
There's non-muscle myosin in skeletal cells as well. So it just happens that skeletal, like muscle cells, have a second special myosin in addition. So, okay. All right. And in our non-muscle cells, actin and myosin form contractile bundles. As you would get into understanding how tissues are built, you would find that there's these adherent belts around the top of epithelial cells that hold tissue layers together. In cells, there's stress fibers of actin filaments that once again help hold cells together. And during the process of cytokinesis, myosin and actin work together to make a contractile ring that pinches cells in two. So this non-muscle myosin serves a lot of roles in holding cells together, and in addition, it's really important for cell motility, which we'll get to next year. So just to go back to a movie that we had talked about, so this is an aplesia growth cone. Well, actually, I'll just ask, what is this big region in the front called here in a growth cone?
Peripheral,
Yeah, the peripheral domain or the P domain. And what is the most abundant sort of molecule or protein found in the P domain of growth cones?
Actin.
Yes, so we've got the P domain here. It's very rich in actin. And this drug cytoskeletal, what does cytoskeletal do? And yeah.
Doesn't it stop the growth on the plus end of the actin?
Exactly. So it's binding to the plus end of actin filaments. It's blocking elongation. And what's the protein that has a similar function in cells?
Is that the capsid?
Yeah, exactly. So yes. So so in this case, cytoskeletal is added, and it blocks actin filament assembly right at the leading edge. So we watch this play through. At the beginning of the movie, there's beads, and the beads are going to be flowing backwards. And then that's the retrograde actin flow. Cytoskeleton is going to be added, and we want to watch this region here as it moves back. Okay, cytoskeleton's added, and you can see that region's moving back. So part of the reason that retrograde flow is happening is through actin assembly, but if actin assembly is blocked and we still have retrograde flow, there has to be more going on there. And that more is non-muscle myosin 2. And we're going to talk about how it works. It turns out there's a drug called blebistatin, and it inhibits myosin 2. What do you think that inhibition of myosin 2 would do to retrograde flow? And I'll throw that out to somebody new. So if myosin 2 is being inhibited, what do you think is going to be happening to retrograde flow? Do you think it would speed up or slow down, or have no effect?
Slow down.
It would slow down, and why would it slow down?
Because myosin is being inhibited, which makes it speed up.
Yeah, so if myosin is really important for this process, the prediction and expectation is if you inhibit it, it's going to slow down retrograde flow. So we'll talk about that. So blebistatin is a drug that inhibits myosin 2 function, and it's really been a really very nice tool for trying to understand myosin. Essentially, it's what's called an uncompetitive inhibitor of non-muscle myosin 2. So it stabilizes it into a transition state. And very importantly, it's blocking force generation, and it's locking myosin in a state where it's unattached to the actin. So we don't have a rigor state here. The actin and myosin can still slide past each other. It's not going to lock everything together. It's just blocking its ability to walk through that cross-bridge cycle. So, okay. So here are two growth cones. So a control's over here, and blebistatin is over here. This cell has been labeled up, I do believe, with rhodamine phalloidin. So phalloidin is a drug that binds to actin filaments. Rhodamine is a fluorescent dye. And we've got these little speckles of actin that we can use to track the flow of actin. And when you look at these movies, what do you see? What are the differences between them? Go ahead.
Retrograde flow is slower on the right.
It does look a little bit slower on the right, maybe? But I will say, does everybody agree with that?
It's hard to tell because like the length of them are different. I don't know if length is the right word.
No. That's a great point. I will say a lot of times, if you just look at these movies, it's a little bit hard to see the difference in speed. And we'll talk about a different way to analyze that in just a second. But you're also right. So there's this big change in the length of the actin filaments. So we won't actually talk about that so much, but it turns out that one of the things that myosin is doing is it's breaking apart actin filaments. So when myosin 2 is being inhibited, there's less disassembly of actin, or it's happening at a slower rate back here, and you get this expansion in the size of the P domain. In contrast, in the cells, you can see that stuff is flowing to this convergence zone, and it seems like it's really rapidly disappearing. So myosin is doing a lot of things. It's generating forces that are pulling things back, and it's also helping actin filaments disassemble because it puts force on the actin that bends and breaks it. Okay, so the velocity. So back in the day, the way that one would measure velocity is you would have a movie like this, you would pause it, you would note a little spot, and then you would go to the next frame and you would try to find that same spot and mark it, and you would step through movies marking little spots. I can say that it was a rather tedious way to get velocity data. And it turns out that a different way of analyzing motion is through the use of kymographs. And this is a kymograph here. And just to kind of walk you through it, it's a visual representation of the motion in the cell over time. one way that I explain chymographs is if you think, if you've got a video, it's really a three-dimensional object. So you have X, Y, and then you have time on the third axis. And when you're watching a movie, what you're doing is you're taking individual images and you're sort of flipping through the stack of images going down in the time axis. So if you imagine you had like a ball that's just sitting in space and wasn't moving at all, as you're flipping through, that ball would just stay in position, right? It wouldn't move. The trick with a chymograph is you can take a movie and you can rotate it and you can look at it from the side. And now instead of having X and Y, you have time and say X on the other dimension. And what that does is it allows you to follow motion in a very nice way. So if we go back to our movie with the ball, if we took that movie and flipped it on its side, so now time was coming down on this axis, what would you see? And I'll throw that out to somebody. Yeah.
Wouldn't that look like a line if it wasn't moving? It would just be like...
Yeah, exactly. So if you flipped it on the side, it would look just like a line here. Okay, so if we have another movie of a ball that's moving across the screen, so let's say that it starts off on this side and then it moves to the other. As you're flipping through the movie, you can see the ball move. Now, if you flip it on the side, what would that line look like?
Like a diagonal.
It would, yeah, exactly. So it'd be diagonal. So it'd switch from having an angle that would be straight up and down to having some angle. And the really cool thing about chymographs is you can look at the angle or the slope of lines in images and then directly measure velocity. So to explain what's going on with this here, so this is a chymograph. We have time on the x-axis, and then we have position on the other axis. Before this myosin 2 inhibitor is added, we can see that we have lines that have a very steep slope. So what that means is they're changing their position really rapidly, and then after blebistatin is added, you can see that the slope of these lines changes, so it becomes much more sort of stationary. And really the wonderful thing about chymographs is that it allows you to take a huge amount of time-lapse data and really rapidly measure how fast things are moving. And this has sort of revolutionized the study of cell motility and cytoskeletal layers. So, okay. So, would we agree that blebistatin is slowing down motion here? Okay. So the question is, if we disrupt actin assembly, what would you expect? So this is a chymograph in essentially the P domain of growth cone. What would you expect to happen when actin polymerization is also slowed down? Yeah, go for it.
Oh, I was just gonna say like a decrease going the other direction.
Well, yeah. So a possibility would be that there would be a further decrease in velocities and stuff like that if these two things are working together. But in what I'm trying to sort of get at is you think, okay, you inhibit myosin, you expect it to slow down, so myosin's making a contribution. You inhibit actin polymerization, if it's making a contribution, it should also slow down. So, okay. And yeah, so this is just a little note on how to understand chymographs. But so here is the entire experiment where both of these things have been inhibited. So just to kind of walk through things, so here is our control over here, and you can see that because it's the line is at a large sort of angle, there's really rapid motion before. And then when blebistatin is added, what does the change of angle mean?
It's slower.
It's slowing down. And then when cytoskeletal is added in addition to blebistatin, what happens? It slows down even more. And then when the cytoskeletal is washed out, which means that the blebistatin, when the blebistatin is washed out and there's just cytoskeletal, what's happening to the slope of the line here? It's increasing. And all of these are sort of, and I guess, what are the implications of these data then?
It means if you want to reduce the speed, you need both leptin
Blebistatin, yeah.
and
Cytoskeletal?
Yeah.
Yeah. So that's at a pharmacological level. So at the cellular level, the implication is that both myosin activity and actin polymerization are both contributing to this actin retrograde flow in the P domain of cells, or the P domain of growth cones in the lamellipodia cells. So yeah, all right. Yes, go ahead.
You said myosin breaks apart actin filaments.
Yes.
Sometimes. So does the myosin, is the actin polymerization kind of dependent on myosin's action in some way?
So myosin assembly is regulated separately from the polymerization of actin. I'm not sure if I understood the question.
Because you were saying that when myosin is working, sometimes it breaks apart the
Yes.
filaments.
Yes.
Thinking whether actin depolymerization is dependent on myosin's action in some way.
Oh, the question is, is actin filament disassembly dependent on myosin 2? And it is very much so. So yes, that is correct. All right. And this is just a quantitative, so we're looking at chymographs, at the pictures, and this is just really showing that, the surprising thing is, if you just inhibit cytoskeletal by itself, it actually doesn't have a major impact on the velocity of actin filament motion. You can see that when blebistatin is inhibited, it really does slow things down. But when you combine both inhibition of actin polymerization and inhibition of the motor, you do see a combined effect. So there's some subtleties here, and there's a little bit more going on, but the big point that I'm trying to get across with just the stuff I showed is that actin assembly, myosin 2 activity work together to drive actin retrograde flow. So, all right. And in terms of what that does is that in combination with molecules that can interact with the outside world, this allows for the generation of traction forces. This is what allows cells to move around the world. So in this particular movie, we have, once again, an aplesia growth cone. The C domain of the growth cone is here. We would have the transition zone, or the T zone, right at the border between the P domain and the C domain. You can see there's nice retrograde flow that's happening in this growth cone. And in this particular case, a bead that is coated with a cell adhesion molecule is placed on the growth cone, and then a pipette is being used to hold it in place. So you can imagine that normally if you would put a bead on the growth cone, it would just be pulled back into the C domain of the growth cone. But here, this is a large bead. The bead is just being held in place with this pipette. And we want to look at what's going on in the growth cone as it responds to this bead that has these cell adhesion molecules on it. So let's watch the C domain of the growth cone. So if you watch the C domain of the growth cone, what's happening with that over time? It's moving forward. So, okay. And then if you look right here, right at the leading edge, what's happening? Exactly. So you see there's just a little bit of a bulge that's sticking out. It isn't nearly as obvious as what's happening sort of in the C domain. And yeah, so the big sort of idea is that, so here, the cell's gripping onto the bead and it's trying to pull the bead backwards. And this generates a reaction force that on one side, we have a lot of myosin 2 contraction happening here at the T zone. And because this actin is being held in place instead of pulling back, then what happens is the cell's using that to pull the C domain forward. And then the second part of this is that, and you can't, I don't think you can see it here, there's also going to be a reduction in the velocity of retrograde flow. So at the very leading edge of the growth cone, actin is trying to assemble as fast as it can, but it's also being pulled backwards. So if it's assembling as fast as it can, and it's normally sort of being pulled backwards, when that bead gets locked into those actin filaments, then the retrograde flow slows. And then what happens is the actin assembly can happen without it being pulled back. And that's going to help push the plasma membrane forward. So the idea, or what I'm trying to show you here, is that when actin filaments can interact with the outside world and generate traction forces, they can pull the world back and they can pull themselves forward through the use of these motors and actin assembly. In terms of the molecular basis, integrins are a really important molecule for cells interacting with the outside world. And just to kind of get you oriented, here's the plasma membrane of the cell. The extracellular space would be towards the top. The interior of the cell and the cytoplasm towards the bottom. Integrins are transmembrane proteins, so they're crossing the plasma membrane. And then what they do is they can interact with these extracellular matrix proteins. So they can grip on and bind in a very selective way. And on the inside of the cell, we've got our actin filaments, and there are a set of proteins, in particular talin and vinculin, which link integrins with actin filaments. So in terms of the way that the cells interact with the outside world, so there's going to be an extracellular matrix molecule, for example, like laminin. Integrins, they can be regulated too, but they're going to reach out and they're going to grip that laminin. They pass through the plasma membrane, and the tail of integrins are going to interact with talin and vinculin. There's a complex of proteins that links integrins to actin filaments, and then these proteins are going to transmit forces to the actin filaments. And as we'll talk about in a bit, talin and these other proteins are thought to be part of what's called a molecular clutch, and I'll talk about that in just a little bit. In a bit. Actually, I'll do it now. So how many people, yeah, what is a clutch? Like, when you hear a clutch, what do you think? Purses, that's, yes, for sure.
Like a car.
A car, so, so yes, so clutches are in car.
Like a clutch of eggs.
A clutch of eggs, for sure. So the clutch here is actually more of a reference to the clutch that's found in the car. Has anybody here driven a manual transmission with a stick? Yes, all right. Okay, so from your experience, so one surprising thing for many people is there's 3 pedals on the floor, right? And what's that third pedal called?
Clutch.
The clutch. And what that's doing is it's actually regulating the clutch that's in part of the engine. And what happens when you push down on the clutch?
Well, that's when you switch gears.
So it disengages the
Yeah.
clutch, so it breaks that connection. So when people were starting to understand cell motility, they were making analogies with how cars work and stuff. And sort of part of the idea is that there's a clutch, and in the engine, what's really happening at a practical level is when the clutch is engaged, there's friction between the engine and the drivetrain that allows the car to move forward. When the clutch gets disengaged, those things move apart so the engine can spin freely and change its velocity of rotation and stuff like that. So this is just kind of the molecular analogy for that sort of clutch. All right, so in terms of the clutch hypothesis, the idea is this clutch is a linkage. And if we have a cell or a case where the clutch is disengaged, that linkage is not there. So if we have a cell that is not moving forward, we can look at this and we can just imagine this is the leading edge of a cell. So the very tip of the cell is going to be over here. We have an actin filament, the plus end's there, the minus end's there. So actin's depolymerizing in the back, repolymerizing in the front, and then you have these integrin molecules attached to the extracellular matrix, but you can see there's no connection between the integrins and the actin filament. So the clutch is disengaged. And putting this all into motion, what's going to happen is that there's going to be rapid retrograde actin flow because you get the force of actin assembly helping to push actin backwards. And then there's also going to be myosin in the back that's also going to be pulling the actin filaments back. When the clutch gets engaged, the situation changes. And now, where there was no linkage, a linkage has been formed between these integrins and the actin filaments, once again through these molecules, tubulin and vinculin. So the clutch gets engaged, and when the clutch is engaged, the actin filament is being locked down to the extracellular matrix. So now, instead of the actin moving backwards, it stays in place. And because it stays in place, the force of actin assembly can push the plasma membrane forward. And then in addition, there's going to be contraction with myosin back here. And in fact, that's going to be pulling the rest of the cell forward as it is pulling the outside world back. So all of that works together to drive the protrusion of the leading edge and the forward advance of the cell. So yeah, okay. So yeah, and then actually I've got a clicker question or two. Let's see what they are. In terms of the clutch hypothesis, what leads to an increase in the rate of retrograde actin flow? So, okay. So we've got our clutch hypothesis here. What would make retrograde actin flow speed up? And here are 4 options. So let's see here. Let's just start at the top. So if the myosin 2 inhibitor blebistatin was added, what would that do to the velocity of retrograde flow? It'd slow it down, and we've already seen experimental data supporting that. So what would happen if we disrupt actin polymerization? It would slow it down, and what's a drug that we could use to do that? Cytoskeletal. And latrunculin is another drug that's very commonly used. So, okay, so that's true. Disengagement of the clutch, would that, let's see, I'm sorry. So what would increase the rate? So would clutch engagement increase the rate of actin retrograde flow? So it, of course, is going to slow it down. So this would be disengagement of the clutch. So when the clutch is disengaged, retrograde flow goes up. Okay. And then just in terms of this model that we talked about, which would lead to an increase in the rate of cell movement.
Engagement.
Clutch engagement, and exactly. So that's the sort of big idea. So that brings together these ideas of the clutch hypothesis.
Yeah, I have a question from the first clicker question.
Oh, sure.
Yeah, I thought like from the graph, disruption of actin polymerization doesn't really do anything at first unless it's coupled with myosin.
It's a little bit more complicated. In other systems, people have seen a reduction in the rate of retrograde flow. I guess, yeah, we can sort of talk about that. All right. This is getting into a deeper bit of a topic, and that is, if you just add cytoskeletal, it doesn't have quite the same effect that, actually, let's see here, that you would expect to see otherwise. And kind of what the thought here is that, yeah, so most of retrograde flow is actually being driven by non-muscle myosin 2, at least in the particular circumstances that these cells were being grown. So the thing that's kind of interesting is that, so if you inhibit bloody statin, what's happening is there's less myosin, active myosin, that's pulling these actin filaments back. And what happens next is that there's going to be an increase in the force resulting from actin polymerization that drives retrograde flow. So normally there's two things working. Myosin is pulling things back, and actin polymerization is pushing things back. So if you stop that pull, then the other process is going to start taking over the job. And it's going to start generating most of the forces there. So you sort of switch from a case where myosin activity and actin assembly are working together, where actin assembly is the sole thing that's driving this process. And it's actin assembly being the sole thing that's driving this retrograde flow. Then, when the system's been reduced to being driven by just actin assembly, when you block actin assembly, it becomes evident that there's a decrease in the velocity. It becomes evident that actin assembly is important for contributing to retrograde flow. And the reason I hesitated to talk through this in the first place is that you almost need to have a deeper understanding to appreciate the certain complexities there. But yeah, it's a good question. I'm glad you asked. So, okay. So with that, this is kind of an introduction to some really basic ideas in cell motility, the clutch hypothesis, how actin assembly and non-muscle myosin 2 work together and are regulated to help cells grow. So with that, I'd say have an awesome day, and I'll see you guys later.